A number of you have come to me asking for a little bit more explanation for this rapid plus minus sequencing technique that was utilized in the um, Michael Smith paper where he was working with site-directed mutagenesis. Now, that question, a few of you were talking about the proteins and the bacteriophage he worked with, but really, the question can be answered by looking at both this slide and the following slide in that same PowerPoint presentation. And so, what I'm going to do in this video is talk about this sequencing technique, how we can interpret it, and hopefully this will give you a little bit more clarity for your exam corrections. So in the rapid plus minus sequencing technique, this was actually a sequencing procedure invented by Fred Sanger. You may have heard about Sanger sequencing, as you can see here, you can see his name. And this was just sort of one of the iterations of how sequencing was completed when we first started doing sequencing. And this particular technique utilizes a two-step elongation process. And so what that basically means is that we take some genetic material, we put it in a test tube, we have some sort of reaction, and then we take that material, put it in another test tube, and have a second reaction. And those two reactions are going to be what is ultimately helping us figure out the sequence of this genetic material. So if we are looking at the first step, that is going to be highlighted in this first half of this image here. And so essentially what was done was we looked at, and now this is just an example of this sequencing. I will say this isn't the actual sequence utilized by the paper, but for the sake of explanation, this will just be fine. So the template DNA would have been that bacteriophage DNA, but here we can see it drawn out here. And just like we do for PCR, there is going to be a primer that is going to anneal to the sequence shown here. And that primer is going to be upstream of the specific sequence we are interested in looking at. Now, in this case, for this example here, we want to confirm that we have a sequence of ATGCTG. And so as we go through this, we can kind of use that as a reference point. So in this first reaction, what we are adding into our test tube is a DNA polymerase, 4 triphosphates, and then some, oops, my highlighting got weird, and one that is going to be P32 labeled. Now, essentially what that means is that this DNA polymerase, which is going to be replicating DNA, it's what we used in PCR, it's what cells use for DNA replication. They are going to be adding this these nucleotides, and we are going to be looking to see which basically see how these templates are being elongated. And so we have our template strand shown here. That's the same thing as we were looking at up here. And then this squiggly line represents our primer that's going to be, elongated. All of these you can imagine are nucleotides here. And then eventually we're going to hit this sequence of interest. And now in this sequence of interest, we are going to have nucleotides that are going to be added to the this elongating strand, and it's going to be extending at sort of a variety of different spots. So we are, in theory, going to end up with new strands that are maybe full length. Maybe we'll get some that are stopping after this A, after this G, the C, the A, and the T. And so ultimately, we can imagine that DNA replication, it's not all going to be exactly the same. It's not all going to be exactly oriented. Um, and we are going to have these nucleotides that are going to be um, 32 phosphorus labeled. So it's going to be an isotope. We'll be able to um, visualize these on a gel, like, once we get to that point. And so these reactions here, they're all going to be at these different lengths, and we are going to eventually force stop these reactions. And so then that way they are no longer going to elongate. And that's why we're going to end up with all of these, different lengths of strands. So we'll have some that are longer, and in theory, we'll have a combination of every different type of length. That's the first step, is elongating the DNA at differing lengths so that we end up with all of these different length template, or I guess I should say complementary strands as such here. And now, what we are going to be looking at is the second step. Now, the samples, so all of this, I should say all of these nucleotides, and I'm going to just go down here for maybe a little bit more of a visual. So we're going to have, all of these nucleotides in the tubes, and like I said, they're all going to sort of be some different lengths. And we are going to take these pooled nucleotide strands and divide them into two separate groups. I give myself a little bit more space. So what we are going to end up with is two groups. One of them will be our plus group, and the other will be our minus group. Now, within these two groups, we are going to break down these into 4 separate tubes. So we are going to just draw out our tubes. And in these tubes, we are going to label them as plus A, plus C, plus T, and plus G. And you can imagine that over on the other side, we are also going to have additional tubes, and we are going to have these labeled kind of in the opposite way. So we're going to have minus A, minus C, minus T, and then minus G. Now, the plus or minus explains what exactly is going on inside of the tube. And so if we have our labels over here, what that essentially means is we are going to be adding some of the differing length of templates. And you can imagine, like, I'm just kind of simplifying this. Know that there are so many copies of all of these. So we have all of our different complementary strands. And then we are going to be adding in certain nucleotides. In the plus A tube, we are going to be adding some adenines. In the plus C tube, we are going to be adding some cytosines. Plus T will add thymines, and plus G will add guanines. Now, over on the other side with our minus tubes, what we are going to be adding, aside from, here I'll draw our template strands. Cool. In the minus tubes, we are actually going to be adding everything but the nucleotide listed. And so what that means is we are going to have in that minus A tube, we are adding C's, T's, and G's. In the minus C tube, A's, T's, and G's, so on and so forth. Cool. So now we have set up our quote unquote plus minus sequencing reactions. But this is not totally done just yet because now we have to run some reactions to actually gather some information. So I'm going to just zoom out and frankly, I'm going to give myself a little bit more space. And we are going to continue on with these sequencing tubes. And so for the plus sequencing, what we are going to have are these template strands that will be all these different lengths. And for the sake of consistency, I am going to record the actual sequence that's utilized example, so then that way everything is consistent. Alrighty. So in this tube, what we are going to have is some sequences that are T, A, C, G, A, C. We are going to have, and you imagine that this is also continuing, so it's longer than what we're just drawing here. We are going to have sequences that go T, A, C, G, A, and then they're stopped. T, A, C, G, and then it got stopped. T, A, C, T, A, and then T. So all of these different sequence lengths are going to be present within these tubes. In our plus reaction, and you know what? Before we even go any further, I'm just going to duplicate this because I know I'm going to want it over here. Cool. Okay. So in the plus tubes, let's just focus on one of these plus tubes and know that all of these tubes are basically going to work the same way, just with a different nucleotide. So in our plus tubes, let's look at plus A as our example. And in fact, you'll look in your PowerPoint slide and see that they also worked with the plus A, so that'll just be helpful. You can compare my explanation to what it is that is discussed in the PowerPoint. So in the plus reactions, not only did they add nucleotides, but they also added an exonuclease. And we remember that exonucleases are going to be cutting down DNA from the end of a strand inwards. It just so happens that the exonuclease that they are adding is a 3' exonuclease, which means that it's going to chop away at the 3' end of the genetic material, which is right here. This is the 3' end, this is a 3' end, so on and so forth. So all of these sort of like staggered ends. And so what is going to happen is that this exonuclease is going to come in and it is actually going to chew back some of the nucleotides. Where the nucleotides come into play, so like for this instance, we're going to be working with our A tube. The exonuclease is only going to chew down until we reach an A. The reason why is because we are going to be able to add new adenines to the sequence, and it'll be adding the adenines more quickly than the exonuclease can actually cut them off. And so what's really going to end up happening is we are going to be able to remove this here C. That'll be removed. Right here, this A will actually be maintained because that's actually already at the end of the sequence. Then, when we're looking at the next sequence down, this G is going to be removed and this C will be removed. So we'll just erase those. Further down, the next row, we'll also see that C removed, that A will be removed, and actually this T will be removed, and you can imagine it would chew back until we reached an A and eventually A. And so what I want for you all to see is that at this point, we have an A at the end of each of these sequences, even though we started out with all different lengths of nucleotide strands. We are now ending up with very discrete selections of nucleotides. So, for example, in this plus A tube, we would end up seeing nucleotide strands that are 1, 2, 3, 4, 5 nucleotides long. So I'll just draw here. We're going to have a whole group of them that are all going to be 5 nucleotides. And then down here, you'll notice that we're also going to have a group that's all 2 nucleotides long. We'll revisit this in just a moment because that is what is going to be showing up on the bands. I'm just going to move this down here so we have a little bit more space for our other side. Alrighty, so we just talked about our plus tubes. Now let's talk about our minus tubes. So what we are going to end up with, I'm going to again scooch these over just a teensy bit. And let's deal with our minus A tube over here, just to keep it consistent. So in this reaction, we are going to be adding, again, I'll put this in parentheses because remember, we don't actually add that adenine. Instead, we are going to be adding the C, T, and G into the tube. The other thing we are going to be adding into this tube is a polymerase. And we know that a polymerase is going to be adding on nucleotides. So whereas in the plus tubes, we have an exonuclease that's cutting down the sequences, over in the minus tubes, we have a polymerase that will be elongating the sequences. And now, the key here is that these sequences will be elongated until we get stuck. We would get stuck over here whenever there is an adenine in the sequence. So we're going to end up not being able to add an adenine where there ought to be one because of the fact that there isn't an available adenine in our tube. And so, if we are looking over here at our sequences, we can imagine that we are going to have, and I know we would maybe have this a little bit longer, so I'm going to just say we don't know what this next nucleotide is here, so we'll just say ... this sequence will be a length longer. Right here, the same thing would be true. We would add this cytosine and then whatever was beyond that until we reached that adenine. The next one down would actually stay that same length because of the fact we know the next nucleotide that would be added is an adenine. The next one down after that, we would be able to add this guanine in place, but nothing beyond that because again, where we're missing is this adenine, so this one would be blank. Down below, what we would end up with, we would add the cytosine, we'd be able to add the guanine, again, stopping where an adenine would be added. And then, what you'll also notice down here is that we wouldn't be able to add anything else to this sequence that starts with a T because of the fact that the next nucleotide would again be an adenine. So what we end up with in our minus tubes, especially and specifically our minus A tube, would be a sequence that is, and for the sake of just explaining this, we're going to just pretend that these first two, we'll just say that there are 6 nucleotides long. We'll pretend like that next nucleotide in the sequence would have been an A. We then have a grouping of 4 nucleotides long. And then we also have a strand that would be 1 nucleotide long. So now what you can imagine is that in our plus tubes, we have nucleotide sequences that are 2 nucleotides long and 5 nucleotides long. In our minus tube, we will have 1 nucleotide, 4, and 6. So what you'll notice is that we are going to be like basically 1 nucleotide away from the other. So over here, we would go from 5 nucleotides to 4 nucleotides. Over here, we would go from 2 nucleotides, and instead we would see a band with 1 nucleotide. Okay. So the very last step of this sequencing is going to be actually running these samples on a gel. And so if we look at this, this sort of like practice band, and I'm going to just zoom in a little bit on the left-hand side here, because I think this does a good job of explaining that. And what you can see based on basically everything I just explained, just to recap, in our minus system, so I guess they have it reversed, but our minus system, we got 4 nucleotides, we've got 6 nucleotides. Over here, 2 nucleotides, 5 nucleotides, and over here we have that, obviously that 1 nucleotide. So those numbers are consistent. Now, when we run this on a gel, what is going to happen is that in our tubes, we are going to have a very discrete banding pattern. And this is going to be apparent when we are loading the samples into these lanes. So, for instance, let's say how are we going to do this? Okay. So I'm going to just draw something that kind of looks like this, just so maybe you can see a little bit more clearly what exactly is happening. And I'm going to draw, okay. So for the sake of this, let's say this is our minus A and this is our plus A tube. And the rest of that is just kind of erase. Alrighty. So in this gel, we are going to have nucleotides of different lengths. So I'm going to say that at like this position, this would be a sequence of 1 nucleotide. This will be 2, 3, 4, 5, 6, 7, 8, 9. Okay. And what we know already is kind of predicting where these bands would fall. And so if everything worked perfectly and our sequence was what we expected, in our minus A column, what we would expect to see would be a band in the first position, a band in the 4th position, and a band in the 6th position, shown here. In the plus column, We would see a band in the second position. We would see a band in the fifth position. And honestly, if we had completed and gone further down the sequence, just kind of based on my like pretend sequencing up above, we would also see a band in the seventh. What you should notice between these two columns is that in the plus A column, we increase the band size by one nucleotide relative to the minus A system. We can also see that over in this image, right actually on our slide. So as we are looking at the columns, and I will add some like highlighting marks, in our minus system, this would be likely representing, I guess, I was going to say this would be the minus A column. And then this would, yeah, okay. So plus A. So let's say that this is going to be like one nucleotide space. This is going to be the two nucleotide space. We also see if we continue up, we have our, oops, this would represent our four nucleotide space. Oh my goodness. And then this other would represent the five nucleotide space. And what this image also shows us is relative, like what the other plus minus lanes would end up looking like. And so in a minus T system, it would technically have a band, based on what we were looking at. It would actually have a band at like the 0 position, and next to it, it would have a band in the 1 position because the nucleotide strands would be either totally cut back beyond what we were before, or we would have one nucleotide here. Continuing on, we would have the same sort of thing for C and same thing for G. And so how these scientists interpret these results is they will utilize essentially a horizontal line, and they're going to go through and look to see, all right, where are we going to be hitting a nucleotide? So if we look over at the minus side first, we're going to be moving our line up along. We're first going to hit a T, then an A C, a G, an A, another C. And that's going to just kind of continue on in that pattern. So if we were to record what bands we end up seeing, we would start with a T, end up with an A next, a C, a G, an A, and a C. Now, in our minus column, we actually are going to end up seeing probably the same sequence, but the nucleotides will be shifted by one. And so over here, we're going to do the same thing. We're going to move up and we're going to get a T first, an A, C, G, A, and C. And as we do that, you'll notice that the sequences are actually going to end up being the same, that we'll get from both our plus side and our minus side. Now remember, the banding pattern of like the nucleotide length we'll end up seeing will be shifted by one position from compared to the minus side to the plus side. And so we are going to see the same patterns on each of these sides. So we're going to have these ones all being in a pattern, and then on the other side we're going to have all of these ones being the same pattern. And as long as those two patterns are consistent, we can feel confident in our sequence of genetic material. And so in the context of this paper, what they wanted to basically determine and confirm was that did we effectively get a change in nucleotides? And that was ultimately what we did end up showing when they were looking at the gels from the paper. So, like, let's say, for instance, we wanted to change a C instead of a G, we would just look for that in the sequencing results. Hopefully this is a little bit clearer, and hopefully this helps understand why things were where they were and why the bands were specific discrete lengths versus such a range, and how we can utilize this technique in order to better understand sequencing.